In this video, we're going to continue on with proteins. And so we'll pick up here where we left off in the last video, looking at the different types of amino acids, which we've basically already done, but we'll just rehash it. Number two, what's the difference between catabolic and anabolic reactions? And then talk about this idea of primary, secondary, tertiary, quaternary structures of a protein. All right, so remember the building blocks of amino acids. There's amino acid number 1. This is amino acid number 2. Okay, so what I'm going to do here, we're going to take amino acid number 1 and combine it with amino acid number 2. Now, why am I doing this? I'm doing this because the building blocks of proteins, the building blocks are amino acids. And all that really means is when you look at a protein, it's a bunch of amino acids strung together. I used these examples of letters before, and it's still useful here too. So what are words? Words are a whole bunch of letters strung together. Okay, and so similarly, what are proteins? It's a whole bunch of amino acids strung together. So let's string together these amino acids. This slide is showing you then again this particular amino acid. It doesn't really matter which one it is. You know by now that R group can be a lot of different things. And another amino acid is right over here. Now, look at what's happening here. We've got an OH from this carboxyl group, and we've got a hydrogen atom over here on this amine group. And what's going to end up happening is that these two are going to hook up and form together, and this carbon is going to dump OH, this nitrogen is going to dump H, and carbon and nitrogen will then form a covalent bond. And so as that happens, there we see it. So if you see, look, this is your first amino acid that we saw previously, and there's your other one right over here. So I just put two boxes around them. This box right over here is indicating what was just over here a moment ago, right before this chemical reaction occurred. And then this box over here on the left, this guy over here, is actually representing, I guess I could color code them, but if you're watching this video, you're watching me drag these, so you should know where I'm at. So this one right over here is this amino acid, this one over here is this amino acid, but you see now that they're bound together. Now this is a covalent bond, it's just your straight up normal run-of-the-mill covalent bond, but because it's occurring between two amino acids, we're gonna also say it is a peptide bond. Now, all these other bonds have names too, meaning that when we talked about polysaccharides, and I told you that glucose can hold hands with the next two adjacent glucoses, or they can link by linking arms, or they can wrap their arms around each other's shoulders. Remember that whole analogy that I gave you before? Those also have specific names for those types of bonds that are formed. I just didn't really care about them. But here, this one is commonly used, like in a lot of regular kind of biology conversations. So what I want you to know is that this thing is called a peptide bond. It's called a peptide bond. And what is it? It's just a covalent bond between amino acids. That's all it is. All right. So now, what did I just do? Let me go ahead and delete these boxes that I just so we can kind of unclutter the picture. And so what we just did is that we connected these two, and I synthesized this peptide bond, and in doing so, I made a dipeptide. That shouldn't be new to you because we talked about monosaccharides hooking up to become disaccharides, and if you add on another monosaccharide, instead of saying a trisaccharide, in that case, we would just call it a polysaccharide, right? In this case, we will go just a little bit higher. I don't know why, that's just the convention people talked about. If I were to string in one more amino acid, there we could call it a tripeptide. And then after that, we'll call it polypeptide. I don't know if those are hard and fast rules or just kind of norms and conventions, but nonetheless, we're going to follow those precedents. Okay, so here we have a dipeptide. Now, this chemical reaction is called a dehydration synthesis. This is really important because you'll see these other places. It's called a dehydration synthesis. And why is it called that? Well, what does dehydrate mean? You know, dehydrate means you don't have enough water. So you've lost water. Well, in the big scheme of things, what I've done here, by eliminating this hydroxyl group and this hydrogen atom, these two end up hooking up together, forming H2O. Right? There's H1, there's H2, and then there's O. So water is released. So during chemistry, true or false, you can make water. And the answer is yes, you can make water. Now, can you make enough water to sustain life? No, you can't do that. I mean, which is why you have to have some water source, whether drinking or through your food every day. You can't go very long without water. But it is true that you can make water. And that's why we say there's a dehydration among these two amino acids that occurred. On the other hand, if you look here, it's a reversible reaction, right? I can build something up, or I can actually break this down to reform my individual amino acids. In that situation, you need water as well, and water shows up, and water gets split, and water ends up breaking these bonds apart. And when water itself gets split, it reforms that OH group, the hydroxyl group, and the hydrogen atom. So this is called a hydrolysis reaction. And that ending here, lysis, L-Y-S-I-S, lysis means to break. What did I break here? The term hydrolysis comes in not because water ends up being split. The term hydrolysis is used here because water is used to split this peptide bond. Okay, so I broke this down, hydrolysis. Okay, now broad generic terms. To build something up is an anabolic reaction. To break something down is a catabolic reaction. You might have heard people talk about the use of anabolic steroids. You know, if you're a high-performing athlete or something like that, sometimes people are using anabolic steroids. You're not supposed to. You shouldn't. They're largely illegal, but they're anabolic in the sense that they're building up muscle mass. Okay, catabolic reactions are breaking things down. It is not fair for us to say one is better than the other. They're just two sides of the same coin. That's life. That's chemistry. We build stuff up and we break stuff down. And so that's what anabolic and catabolic reactions are. Okay, so let's see where we're at. We talked about the 20 different amino acids and the differences, the R groups. We looked at the difference between catabolic, breaking down, and anabolic, building up reactions, specifically looking at hydrolysis and dehydration synthesis. Now we got to talk about this part over here, primary, secondary, tertiary, quaternary structures of a protein. Okay, so let's take a look at this slide over here. You know that spelling matters, and what I mean by that is what I'm trying to do here is I'm trying to go back to this analogy that I used before where I said words are a bunch of letters that are strung together. Now they're strung together into meaningful, coherent patterns of organization. So S-T-I-C-K means something different than S-T-O-N-E. So even though they may have a couple of the same letters, in this case, the rest of the letters really, really do matter. So of course, that's obvious. Everybody gets that. So what I want you to see then is that when we string together amino acids to make a protein, because that's what we're going after here. We're trying to string together amino acids to make a protein. Stringing together amino acids, let's just make this up, and let's say stringing together glycine, alanine, valine in position 1, position 2, position 3 is different than stringing together alanine, glycine, and then valine, right? So the order in which we say things does matter, and you know that. Let's say these were letters. Let's just say this was W, E, and D. Well, you know that if I did W-E-D, that spells wed. And those two were wed last weekend. They got married, yay. Versus D-E-W, there's morning dew out on the grass. And so, you know, they're way different things. Same letters, but different organization means something different. On the other hand, if you just substitute one out, H-A-T means hat, of course, that you put on your head is something different than a hut that you might live in somewhere. And so swapping letters out really does matter. Well, the same applies to proteins. So to say it differently, does the spelling of a protein matter? Do we care? Maybe not us in our class specifically, but do chemists out there and molecular biologists care about the way a protein is set up, which amino acids they use and which positions they occupy? And of course they do. Now, I might use language that says something like, the first letter is S, the second letter is T, the third letter is I, the fourth letter is C, the fifth letter is K. Whereas in a chemist, they might say something like the first amino acid is this one, the second amino acid is this one, or the first position is this, second position is this. But my point is, thematically, it's the same thing. Spelling matters. Certainly it does. And so what we want to know then is that when we start talking about these other components of a protein, we want to see that, well, all amino acids are connected together. And when they're connected together, it's going to form a jumble of letters. And at that point, we're not a full-blown protein yet. We're a polypeptide, many peptides. We're a polypeptide. And so when people describe different parts of the protein, there's different descriptions to the protein. The first is, okay, what's the spelling of the protein? You know, which amino acid is in the first position? Which amino acid is in the second position? Which is in the third? So on and so forth. And when we talk about that, what people are referring to is the primary structure, okay? So nobody in the world is going to say, what is the spelling of a protein? But what they really mean, what they'll ask you is, what's the primary structure? And that is simply the sequence of amino acids. Now, what I want you to see is once we have that down, the amino acid itself must fold. And you know, as I look at the slide, I realize perhaps this sentence is better positioned somewhere like that. Maybe that's what it should be. Okay, so really, the primary structure is just the sequence of amino acids, but from here on out, these other things are what are when the polypeptide itself folds. Now, let's look at these things called secondary structures. When you look at a bunch of amino acids, so if you look at something like this is actually an amino acid, which is bound to the next amino acid, which is bound to the next one, so on and so forth. If we were to unwind this ribbon, then you'll see that it's just a string of amino acids. And you can kind of make that out over here. This is a slightly different structure, but it's a string of amino acids. What I want you to see is that when you string together a bunch of these things, the strand of peptides, the polypeptide itself, is going to start to fold. It's going to start to change shape. You're not going to be able to lay it out in a straight line. And so there are two stereotypical ways that small segments can fold. Now, let me be clear. If you string out, let's say, 100 amino acids, I'm just making up that number, I'm not yet talking about the whole thing from position 1 to position 100 folding. What I'm talking about is small local regions, let's say position 35 to 45, and then at position 72 to 86, something like that, small regions of that entire strand. So what we're looking at here, as amino acid 1 is way over here, and if you go all the way down, let's just call this amino acid number 100 down the road. So at certain regions, you'll see that certain sections take on characteristic folding patterns, and those come in two flavors. Either it forms like this kind of curly Q thing, or it forms an accordion-like structure. Those are the two famous characteristic secondary structures of a protein. And so this curly Q is actually called an alpha helix. So an alpha helix is a famous secondary structure of a protein. And the other thing, this accordion-like structure, is called a beta pleated sheet. So again, what is the definition of a secondary structure? The definition of a secondary structure are regional foldings of the polypeptide chain. And those regional foldings take on two characteristic shapes. It's either an alpha helix or a beta pleated sheet. This is just from another book showing you there's my string of amino acids, and I can describe the spelling or the sequence of amino acids as the primary structure. Then regionally, I might see some of those amino acids start to kind of curl around this way, and they do that based on the charges on the R groups primarily, based on the distribution of charges. What I want to be clear on then is that any given protein, if you make that protein 100 times or 1,000 times, it's always going to be the same regions that fold in this characteristic patterns. And whereas other parts are going to always and every single time fold in these characteristic patterns. So again, this is the alpha helix and this is the beta pleated sheet. What is the tertiary structure of a protein? The tertiary structure of a protein is when that entire polypeptide chain now folds up on itself. So remember the example that I told you. I said that, okay, amino acid number 1, number 2, number 3, all the way down to amino acid number 100, which here is valine. Regional foldings can take on that alpha helix appearance or a beta pleated sheet appearance. But then what ends up happening is the whole darn thing starts folding up on itself. And you don't see that so much over here, but maybe on this picture you can see that now here, the whole polypeptide has now folded itself up. Now, tertiary structure in this definition is a three-dimensional shape of the polypeptide chain. And if you were to zoom in, yeah, you'd see an alpha helix right over there. Ooh, I see another alpha helix right over here. I see a beta pleated sheet right over there, kind of embedded within this entire wound, smushed up tertiary structure. So tertiary structure is when the entire chain folds. Now, for many proteins, we're done at that point. At that point, we're finished, and many proteins do not have a quaternary structure, but there are a handful that do. Now, what is a quaternary structure? Quaternary structures exist only in some proteins, and that is when 2 or more polypeptide chains are involved. So what that means is polypeptide chain number 1 has its spelling, there's regional foldings into 2 characteristic shapes, then the whole darn thing folds up on itself. And then, over here, actually off screen, I suppose, another polypeptide chain would do its thing. And then those 2 polypeptide chains, here it's indicated by blue and then the gray one. Each of those, both of those, I should say, come together and now they form one protein. So this protein has a quaternary structure because it has 2 polypeptide chains. Now again, if you were to look, do each of those polypeptide chains have a tertiary structure? Yeah, they do, right? If we could take this gray one and snap our fingers and make it disappear, what you would see is just this blue one remaining right over here. So that's got a tertiary structure. Now let's bring the gray one back. The gray one also has a tertiary structure. Do each of those polypeptide chains have alpha helices? Yep, there's an alpha helix there. Oh, there's an alpha helix there, right? And a beta pleated sheet here, beta pleated sheet right over there. And again, any polypeptide chain may have more than one area of an alpha helix, more than one area of a beta pleated sheet. And then, do each of these have a spelling? Yep, this polypeptide chain has a spelling, primary structure, and this polypeptide chain has a spelling, primary structure. Okay, so those are proteins. Probably the most famous protein with a quaternary structure is hemoglobin, and you will look at that when we get into Bio 142, where we look at blood. All right, let's wrap it up. 20 different amino acids. The difference among all those is the R group. The difference between catabolic reactions, which break things down, and anabolic reactions, which build things up. Now you understand. And now you know that primary structures are the spelling of a polypeptide. Secondary structures are regional foldings that take on two characteristic forms, either an alpha helix or a beta pleated sheet. You know that the tertiary structure is when the entire polypeptide chain, with its embedded secondary structures and all, folds up on itself. And quaternary structures exist only in some proteins, and that's when two or more polypeptide chains that each have primary, secondary, tertiary structures have now come together. And those are proteins with a quaternary structure. And that'll wrap it up for proteins.